cave did not remain passive. When the instruments moved deeper, the environment changed. Gas levels shifted, biological structures reorganized, and the system responded for the first time in millions of years. This is not a separate discovery. It is what happened next. In Part 1, explorers opened a cave sealed beneath Antarctic ice for millions of years. What they found challenged everything we understand about life in isolation. But deeper inside the cave, the rules began to change. If you haven't seen Part 1, you should, because what you're about to see only makes sense once you understand how this world was first exposed. A pulse of light in a world that has known only darkness for millions of years. This single, placid animal instantly transformed a geological expedition into a biological event of the first order. The rules had changed. This was no longer merely an exploration. It was a first contact with a chapter of Earth's own history we never knew was written. The creature, designated Specimen Alpha, was not alone. Subsequent surveys using low-light sensors and robotic probes revealed a sparse but definitive ecosystem. Stationary filter feeders akin to the giant tube worms formed the base, the primary producers converting the cave's chemical whispers into biological matter. Alpha and a handful of similar slow-moving drifters occupied a higher trophic level, likely grazing on microbial films or perhaps the juvenile forms of the filter feeders. Their biology is a study in radical efficiency. Initial scans suggest a complete absence of eyes or any complex sensory organ dedicated to light. Why would there be? Instead, their translucent skin is laced with a dense network of cilia and chemical receptors, sensing the faintest currents and molecular trails in the stagnant water. Their bioluminescence is not for communication, but a byproduct of waste elimination, a slow-cold chemical reaction that neutralizes toxins. That faint pulse was a breath, a heartbeat. But the true profundity of this cave lies not in the animals we can see, but in the molecular machinery that builds them. All known life on Earth shares a common blueprint: DNA and RNA for information, proteins as molecular machines, a lipid membrane for the cell. It is a system so universal we assumed it was the only system possible. The initial genetic sequencer in the cave, designed to recognize known terrestrial DNA, returned fragmented, confusing reads. It was assumed to be due to damage or novel gene sequences. Samples were flash frozen and rushed under the highest biosecurity protocols to a specialized containment lab. There, under the most stringent analysis, the anomaly revealed itself. The organisms possess a double helix structure for information storage, but it is not built on the same backbone as ours. Our DNA uses a phosphate sugar chain. The cave life uses an alternative molecular structure, one that is far more stable in cold, alkaline, and energy-poor environments. This is not merely a new branch on the tree of life. This is a second tree, grown from a different seed. The implication is staggering. It suggests life arose on Earth not once, but at least twice. Two separate genesis events. The life we know conquered the sunlit world. This other shadow biosphere found its refuge in the deep, cold, mineral-rich recesses of the planet. It has been here, isolated and evolving along its own path, since the very beginning. Antarctica's cave is not just a sanctuary, it is a relic of a lost biology. One leading theory, the cold Terra Genesis hypothesis, posits that while our life likely began in warm, sunlit pools or hydrothermal vents, a parallel genesis could have occurred in icy, subsurface niches rich in clays and metals. Here, the slower reaction rates and stability would favor a different, more durable molecular architecture. For billions of years, these two biospheres may have existed in parallel, one utterly ignorant of the other, until the glaciation of Antarctica finally sealed the last remnants of the shadow biosphere away. This discovery forces us to reevaluate every assumption about the search for life beyond Earth. If two fundamentally different types of life can emerge and coexist on one planet, then the galaxy may not just be filled with life. It may be filled with types of life, each operating on a different biochemical logic. Our probes, designed to find life like us, may be blind to what is right beside us. But with this revelation comes a profound and unsettling responsibility. We have opened the seal. Light, sound, and foreign microbes from our world have now entered theirs. Their immune systems, if they have any, evolved in a world of sterile isolation. A common cold to them could be an apocalyptic plague. The research station is now a maximum containment zone. The cave entrance is under a double airlock. Every scientist who descends undergoes a decontamination more rigorous than any used for spacewalk preparation. The guiding principle is not merely study, it is quarantine. We are not just explorers in that cave. We are intruders in a world that has never known an enemy. The final, and perhaps most haunting, analysis came from the rock itself. Isotopic dating of the mineral layers that formed over the cave's water pool indicates the system has been isolated for approximately 33 million years. This dates the sealing to a period just after the Eocene-Oligocene boundary, when Antarctica cooled dramatically and began to develop its permanent ice sheet. This time frame is critical. It means the life in this cave was trapped after the extinction of the dinosaurs, after the rise of mammals. It has been evolving in absolute isolation while, on the surface, primates arose, stood upright, and eventually built the machines that would find them. The samples from Specimen Alpha now sit in a supercooled vault. The leading xenobiologists in the world are analyzing its biochemistry, not to see how it is like us, but to understand how it is different. Each molecule is a clue to a second genesis. The preliminary report concludes with a statement that is neither celebratory nor fearful, but simply awestruck. The system is not just alien in environment, it is alien in essence. It represents a fundamentally separate experiment in the construction of living matter. Its value is incalculable. Its fragility is absolute. Our primary duty is no longer to understand it, but to ensure our understanding does not become its end. We came to Antarctica to study ice, to understand climate. We drilled through time, expecting to find history. We found, instead, a mirror, showing us a reflection of life that is utterly, profoundly other. It reveals not only the tenacity of life, but its boundless potential for variety. We are not alone on this planet. We have never been alone. We have simply been ignorant of our neighbors, who have been here, in the quiet and the cold, since the world was young. The cave is sealed again, not by ice this time, but by human choice. The lights are off. The darkness has returned. And in that perfect ancient dark, a pale blue-green pulse beats on, a silent heart in the deep, keeping a rhythm we are only just beginning to hear.